Hello, everyone, and welcome to the introduction to my lab, the Carpenter lab. So I'll get right into it. So we primarily work on transcription factors and how those play a role in the process of cancer metastasis. So one aspect of transcription factors that we're quite interested in is the upstream regulation of these proteins. So that includes, several different factors that can regulate transcription factors, either through post-translational modifications, such as phosphorylation, regulating the localization of transcription factors to the nucleus or to its binding DNA binding elements, as well as interaction of these transcription factors with cofactors that either enable or allow for these transcription factors to do their job of regulating genes. So one example of a study that we've done recently on this topic is that we found a pretty interesting uh, phosphorylation profile of heat shock factor 1 or HSF1, which is normally involved in this heat shock response that you can see here. Where we found that AKT1 kind of uniquely phosphorylates a set of residues on the HSF1 protein, and that these residues, phosphorylation of these residues, actually enable different steps of the activation process for HSF1. So phosphorylation at this residue here and this heptad repeat domain is responsible for the trimerization of HSF1, and without that event, trimerization is significantly inhibited. Whereas phosphorylation at these three other sites enable gene activation by HSF1 by recruiting general transcription factors like CDK9 and TF2B to allow for transcription to happen. So in addition to studying kind of the upstream protein regulation of transcription factors, we're also interested in what they're doing downstream. So what are the genes that the transcription factors are binding to? And then how do these genes and which aspects or which of these genes actually enable for cancer progression and cancer metastasis? So one example of a study for this is one involving metastasis. So just a very quick overview of this process of metastasis. You have a primary tumor, and it has to be able to get into the circulation and get out of the primary tumor. And then it will circulate around the body and seed a new tumor in a secondary organ. And this is the overall process of metastasis. And this process of metastasis, and particularly the secondary tumors, is what actually makes cancer so lethal, because the vast majority of cancer deaths are caused by metastasis. So one study that we did is also involving HSF1, is we took cells that are either control cells or cells that have knockdown of our HSF1 protein, and we inject those cells directly into the heart of mice. And so that allows for them to essentially directly enter into the circulation phase of metastasis, so that then they will go throughout the body and form metastases, which you can see on this mouse on the left. All of these bright spots are different metastases after we do that. But when we lose HSF1, we can see that process is severely abated, and that the metastatic burden is much less when we don't have HSF1. So now we're trying to study what aspects of colonization actually initiate the activation of HSF1, so this upstream regulation, specifically in the context of colonization. And then also we're going to be using single-cell RNA-seq from in vivo tumors similar to this to understand what are the genes within the cancer cells that HSF1 is upregulating that enables the colonization to happen. So we're going to be combining the single-cell RNA-seq with ChIP-seq data that we have already for HSF1 and what genes that it binds, and using that to really help us draw a conclusion about what are the genes essential downstream of HSF1 that enables colonization. So those are two examples of some studies, but we have a lot more projects to be working on. So one is related to this transcription factor, HSF1, and trying to understand its role in cancer stem cells, because we have data for that, but we don't know the mechanism and whether we could actually potentially target this pathway as a way to block cancer stem cells. We also have found an interaction between HSF1 and c-Myc, another transcription factor in ovarian cancer. So we have some pretty interesting both basic understanding of the transcription factor as well as translational studies to do with that. The role of HSF1 in arsenic-induced bladder carcinogenesis. We have a model set up to mimic arsenic-induced bladder cancer specifically. And we know that HSF1 seems to be involved in that, as well as something I just mentioned, the role of HSF1 in metastatic colonization, but also the role of HSF1 in metastatic circulation as well. Another transcription factor that we work on is truncated GLI1. So this is an alternative splice variant of the GLI1 transcription factor in the sonic hedgehog pathway. So there's a lot of really big open questions about this because it's fairly new. And one would be the DNA binding specificity of TGLI1 versus its parent GLI1, because they do seem to regulate different genes, but we don't exactly know how. And many of those genes that TGLI1 regulates enables tumor progression. So we also want to study specifically the role of TGLI1 in medulloblastoma, because there is an entire subtype of medulloblastoma focused on alterations in the sonic hedgehog pathway. We also know that TGLI1 expressing CNS tumors can induce activation of astrocytes, and so we want to study exactly how that happens. And we also know that TGLI1 promotes the cancer stem cell phenotype as well, and so understanding the mechanisms for that is also of interest to us. So that is the big overview of our lab. Here's my email if you'd like to contact me with any more questions. I'm looking forward to meeting everyone during the interview process.